Welcome. Today, my colleagues and I will be presenting on the use of unsupervised clustering techniques to distinguish helmeted hornbill vocalizations. Both male and female helmeted hornbills are territorial and emit loud calls that are heard over many kilometers. Vocalizations were recorded via passive acoustic monitoring, or PAM. PAM is a non-invasive approach that relies on autonomous recording units and does not allow for initial identification of calling individuals because data is collected in the absence of human observers. We use the acoustic data from 9 autonomous recording units spread across 3 kilometers squared in Danum Valley Conservation Area in Sabah, Malaysia. Acoustic data was recorded over 2 months of continuous data collection. Our goal was to use unsupervised clustering techniques to investigate the tendency to cluster in our dataset of hornbill calls. We expected to see multiple clusters that would perhaps correspond to different individuals or to male and female hornbills. Helmeted hornbills are a critically endangered species, and gathering information on the population structure, density, and distribution can be used for accurate conservation planning. This is a helmeted hornbill call represented in a spectrogram. A spectrogram is a visual way of representing the signal strength or loudness of a signal over time at various frequencies.
As seen in the spectrogram and as described by Elliot Hamoff's spectrographic analysis of the loud calls of helmeted hornbills, loud calls are comprised of 2 parts, the introduction and the cackle phrase. Our analysis focused only on the notes in the cackle phrase. We compared 2 feature extraction techniques in our data analysis. First, we looked at 12 different features from the spectrograms, including call duration, number of notes, and note rate, duration, and minimum and maximum frequencies. We used Raven Pro 1.6 to estimate the features from the spectrogram. We also used Mel frequency cepstral coefficients, or MFCCs, a semi-automated feature extraction method. MFCCs were originally developed for human speech recognition, but have been shown to be effective features for distinguishing many different types of animal vocalizations. We use the R package TuneR to estimate MFCCs. To test for the tendency to cluster in our dataset, we used an unsupervised random forest algorithm. All analyses were done in the R programming environment. For both sets of features, we found evidence for 2 unique clusters in our dataset. Figure 1 corresponds to the feature extraction for the 12 different spectrogram features. Using the data visualization technique UMAP, or Uniform Manifold Approximation and Projection, there appears to be 2 clusters in the data. Each point represents one of the 204 hornbill calls that were analyzed in this study, and the colors indicate cluster assignments by the unsupervised random forest algorithm. Figure 2 shows box plots for the 12 features from the spectrograms, where the colors indicate the cluster assignments by the random forest algorithm. The central line represents the median, while the box represents the interquartile range. The box plots show that the number of notes, along with some of the spectral features, including note frequency, differ between clusters. Figure 3 corresponds to the MFCC's feature extraction. Similar to the feature extraction with the 12 spectrogram features, we see 2 clusters in the data using UMAP.
Although we did not find evidence for many clusters in hornbill calls, unsupervised clustering techniques worked well in distinguishing between calls of individual northern gray gibbon females. We did see that the fundamental frequency of the first cluster was lower than in the second cluster. The 2 clusters may represent male and female vocalizations as female hornbill calls at a slightly higher frequency.
To continue with the goal of identifying individual hornbills, we will identify groups of calls that occurred on spatially separated recorders that we can consider as independent to test supervised classification approaches. We will also collect vocal data from hornbill individuals in collaboration with our Malaysian colleagues in order to further investigate vocal individuality in these animals. Identifying the distribution of male and female hornbills could be helpful in tracking population structures, and identifying characteristics of an endangered population can be used for accurate conservation planning. Thank you for watching our presentation, and special thanks to the Cornell Lab of Ornithology for funding our research this summer.